Like 3 days.
All right, so DNA replication. So the reason that this is even important is, are you a multicellular organism or a unicellular organism?
Is that a real question?
Yes.
We should know that. You're multi.
And everybody in YouTube world will know too. All right, so multicellular or unicellular? Multi. Okay, so, but how did you start at conception? How many cells were you made of?
23 and 23.
One cell. No, you start off as one cell, you by yourself, because sperm and egg combined, right?
Yep.
And so when that sperm fertilized your egg, or not your egg, your mama's egg, it made you. So what DNA replication is if you only started off as one book of DNA, you only had your one copy of your genome, then you've got trillions of cells, and each one of those has to have the exact same copy.
Out of any chance,
Okay?
like what chance are we being born out of the other sperm cells?
None.
That's
Don't
what
worry
I'm
about
saying.
it.
There's so many, so how it feels like that. You beat the Y'all get what I'm saying?
As they're relevant, okay? So because if we have a lot of background noise, you're not going to be able to understand this, okay? And you're still going to be held responsible. I don't care, okay? All right, now, so going back to that, conceptually, if you don't understand this, then none of this is going to make sense. So if you started off as one fertilized egg, a zygote, you had your unique copy of your genome right there. You got 23 chromosomes from one parent, 23 from the other. This could be this tree out here. It got half of its DNA in its pollen, half in an egg, and it made that willow oak tree right outside the classroom. Same thing happened with them. It's not just with people. So in order to be able to pass your genes on, that same copy of every single new cell that you make, you have to copy DNA. So copy the DNA in a body cell. So every new cell will have its own copy. So its own copy. And if I had enough room, I would have written it out that way. All right, so Wesley said a minute ago, when does this happen? So when does this happen? Prior to cell division and interphase, okay? So when, I'm going to add to the list. Just a sec. Okay, so where does this happen? Where's your DNA locked up in your body cells? The nucleus. All right, so we're going to put nucleus of eukaryotic cells. What if you're a bacterium?
Yes.
Where's your DNA floating around if you're just a one-cell little bacteria? Cytoplasm. Right. Prokaryote. But like they said on that video, this right here is what they're going to want you to know. That wherever the DNA is, that's where this happens. All right, so why every new cell must have its own copy of DNA. So think about your DNA now. Yes, it's your blueprints, but it's also all the instructions for that cell to know what to do. So if that instruction book changes, then it can't tell the cell accurately what to do. That's a mutation. So why every cell must have its own copy. Cell must have its own copy of the genome. All right. And I'm going to put here the genome equals your recipes or blueprints or instruction manual. All right. So that's what I was looking around for. So this genome book, you got one copy. Here's your master copy of your genome at conception. So we're going to draw that in just a second. Okay.
Mhm.
You did get half of that instruction book from one parent, the other half from the other parent, and you put it together and you've got a complete set. So this whole thing has to be copied so that it can pass it on to the new cell that's made. Okay, so when, like Wesley said a minute ago, during interphase, which right now doesn't mean a whole lot, but it will later, because in our next unit, it's called growth, development, reproduction. We're going to get into some pretty fabulous conversations. And we're going to talk about all kinds of neat stuff that really is relevant to your life and your future. And part of it is you'll understand interphase. You'll be like, oh, yeah, well, that's why this happens. Everything will start to really come together. So during interphase, prior to cell division, what does prior mean?
Like before.
Right. All right, so in 24 hours or less, one of your skin cells will actually go through DNA doubles and it splits into two more. So if I have, y'all know that all this is dead skin, right, on the outside.
Yeah.
Even if you exfoliate, it's
Like
pretty
in,
dead.
Like in a bathtub, if you take like I don't like just saying like you can see your dead.
You'll see dirt, grime, soap scum, and it'll have even some skin cells, right? But here's the thing. So let's say that you scratch yourself or you've got a wound. All those cells around it have to do the dividing in order to heal. So everyone, DNA doubles, they split. DNA doubles, they split until they fill in the gap. So that's what we're going to show. We're going to show the conception part first. So let's draw a sperm and write 23 in the nucleus of that sperm. Okay, so human sperm is going to fertilize a human egg, which also has 23 chromosomes. And when it comes together, so there's fertilization, you were a zygote with how many total chromosomes?
1.
How many total?
Oh, 46.
DeAndre,
Wait.
you got to stop the chatter, please.
40, was it 46?
46, add them together. Now the question last period was, did somebody ever not get enough or get too many? Absolutely. In the next unit, we're going to talk about Down syndrome and Prader-Willi
They
and
have their
Klinefelter syndrome and Turner syndrome, all these different syndromes that are created because there's a weird number of chromosomes that just happen, and then we get different products. All right, so this zygote is a fertilized egg. Now, right now, you are the size of a tip of a pin. All right, hold on, you are the tiny, tiny, tiny. You're not tiny, tiny, tiny anymore. You're made of trillions of cells. So what had to happen to this DNA? Before the cell could divide,
It had to add up.
what did it do?
Make a
Make a copy. So literally, this is it. Make a whole copy, and now when you get a copy, how many chromosomes will be inside of there? Not 46. We copy it, multiply by 2. What do you get?
92.
Right.
Oh. I'm not doing math. I was late.
All right. And now each new cell has 46.
You gotta write that?
Okay? So yeah.
Anything she writes, you write. Okay, first of all.
Anything she writes, you write, not she writes.
All right, so let's go back to conception real quick before we draw this thing out. So if at conception, this is what happened, which it did, then these double and they get 4 cells in a ball. These are stem cells. Okay, you are a little ball of cells. What if these two actually became apart from each other? What would be created?
Twins.
Identical twins. So that's why one genome, now they have the same genome.
Say that again. How to
Okay, so if these two cells, they're stuck together, okay, then DNA doubles and they split, and you were just a little ball of cells. But what if these two cells actually split apart totally, you've got two people who have the same copy of DNA.
That's scary.
And that's how you get, that's how you get clones.
That's low key scary. Think about it.
Yeah.
So what you're telling me is
Wait, hold on. RJ asked a question. Hold on.
Dang, they all look the same.
Okay, so this was a good question. So with multiples, what can happen with multiples is most of the time when you've got sextuplets or twins or like quintuplets or any of those, you have multiple eggs that were fertilized by multiple sperm. One egg, one sperm, one egg, one sperm, one egg, one sperm. But they can split, and then technically those could split too. So when you have a multiple, you might have identicals and fraternals in the same group.
That's scary.
It's wild. Yeah. All right.
I think my phone is too heavy for this.
All right.
So I want to bring it
Okay. Now let's go back to. I keep saying cell division, cell division. That the word for that is mitosis. Okay, so we'll get into that in the next unit. You don't have to worry about that right now. Just cell division is good enough. All right. So here's how. All right. Number one. There's 3 steps. And
wait, so when
grab
they do
your
split, do
grab
they
your
have to
blue.
become identical, or is this
These would be identical.
because it's the same number?
Yes. Now, if it's fraternal, there were an egg and a sperm. There was another separate egg and a separate sperm. And that's why their genomes are not identical. They might read almost the same because they came from the same mama and daddy, but it's going to be not quite. The genome won't be identical to this.
But will they look like identical twins? They look exactly like. So if they were born at the same time, they're still gonna look alike?
It's not the same time as not just where the DNA comes from. If it's the same DNA, then they are identical and have the same traits.
Because some twins, like not identical twins, but twins, they'll look alike. Like they'll look alike.
Right. They just have similar genes. Yeah. All right, so here we go. Grab your blue. All right. So in between, what we're going to draw is actually the model that you made. Okay, but we're going to make it a little bit longer, and we're going to write A, T, C, G, C, A. Let's say T. Okay.
Oh,
So what is the complementary? That's another word. Yeah. All right, Jenny, let's get it together. Or you're going to sit right back there. And you and Evan can play whatever you want to play with each other later.
why are you guys so negative? Things like that.
I don't know what you're talking about. All right. Okay, so what is the complementary strand? What is the complementary strand to this strand here? Complementary, not what's identical, but what's complementary. So what would you find across from an adenine? A T. A T. All right, so I'm going to put 2 dots just because it's easier for me. And then we'll put a T. But what do those dots represent? You can put lines.
The hydrogen bond?
Yes. All right. So I'm going to do dot dot A because still same partner, just in different order. Same hydrogen bonds, 2. Okay, but how many go between C and G? 3.
Why?
Just because that's how it is. So on your models, that's how you can get extra credit, is to put the number of hydrogen bonds. Now, you didn't have to, because let's say this is my model and I present today and I say these little lines here are the hydrogen bonds, but I don't have the correct number. That's fine. The correct number just gives you extra credit points. But if you say the hot glue in the middle or whatever the paper clip in the middle, I mean, whatever it is, that's your hydrogen bond. So those are my little dots. All right, so tell me what's the complementary, the rest of it. What goes here?
C, G, C, A.
All right, I'm gonna put those. Dot, dot. All right, yellow. Okay, so like what DeAndrea told us 3 times earlier was DNA helicase does what to these 2 strands?
Zip, split, like zips, unzips.
Splits them, unzips them. So tell me, that's what I'm going to write down. I'm going to say that's what this is, the unzipper enzyme, DNA helicase. How do you know this is an enzyme? By looking at its name. The ACE, right. So DNA helicase enzyme. I wouldn't write down enzyme if you know that ACE means enzyme. Okay, but if you are not, if you're afraid you'll forget, just add the word enzyme. So DNA helicase enzyme unzips the 2 strands by breaking what?
The hydrogen bond.
Very good. Now, are y'all okay if I put just H bonds?
Yeah.
Do you remember that
Yes.
hydrogen? If not, write the word hydrogen, okay? But I'm just going to put H bonds just because I'm here at the end of my board. Okay, so that's the first step. We can't copy it unless we split them apart. So that's what we're going to show in this next picture. We're going to make it look like a Y, okay, on its side. And this is where we're going to end up needing the other 2 colors too. All right, so let's take the blue, and I'm going to keep it like this and go out because I'm going to put the A and T right here. Everything else is unzipped, okay? So this will be T and A. I'm going to make it look like that, like it's. So we've taken, this is your zipper, we're going to unzip your zipper, and now it's like this, okay? So the, you know the teeth of your zipper, you know where they fit together?
Yeah.
That's the hydrogen bonds. Okay. All right. So I'm going to write the rest of this out here since we have it now unzipped. So I've got C, G, C, A, T.
Wow. That doesn't connect, right? It's not gonna connect. Okay, so I'm sure. Yeah.
Yep, they're just separated like that. Now, 6 feet long, this would take a long, long time. So this does not happen at one end and it goes on like that. It actually happens at multiple places along the way. So it happens at, it's a lot quicker that way. And so your DNA can replicate kind of fast. And that way the cell can go ahead and do what it needs to do so it can divide. Okay? All right, so now tell me, what are the complementary nucleotides that are going to be added? What's going to go here? What matches with G?
C.
I mean C and G, right? Okay, so the enzyme that does this, and we still have some stuff to be unzipped. So I'm going to put the helicase is still here doing its job. It's going to go that way. I'm going to use my green, and that's the other enzyme. That is the copying enzyme. That's the one that's actually going to write in, and it's going to fill in and add the nucleotides in the right order. So that's a green, and that's going to be green. It's going that way, and this one's going this way. All right, so because it's the new strand, this is going to be our red strand. So tell me what to put here. Y'all said G. All right, what comes next? C.
G, T, A.
And that is a new strand. So we're going to connect it with the red. All right, how about on this side?
C, G, C.
All right, good. And we're going to go that way.
Oh my God.
I'm telling
Now, what do y'all notice about those two strands?
The
Blue and the red and the blue and the red.
they're identical.
They're identical. So as long as there's no mismatches, and have no mutations, so it can go in and it can put a C here. But there is a spell check enzyme that's going to go there and go around and look for mistakes. But here's the thing. If there were no mistakes, we wouldn't have the genetic variation in this room. And I want you to think about that.
So everybody was the same.
All humans would look the same. So we wouldn't even have straight hair, curly hair. We wouldn't have red hair, black hair. We wouldn't have really tall and really short. All those are changes.
Hey,
Wouldn't have it.
we're gonna be right. It's not fair.
Why
couldn't we all be the same?
Oh, God.
Fair. Nobody was just bullied. Every single day of their life. You know what? I hate school.
You want school makes you.
I know it does.
Okay, so tell me what to write next. So RNA polymerase, that's the green. DNA polymerase copies each strand by adding, do we say identical nucleotides or complementary?
Complementary.
Right.
I was listening. I never listened. One big brain mom.
And this is really just nitrogen base is what we're talking about. So what we need to, I think everybody gets this, but as people present their models, what's going to be different from everybody's model? I'm not talking about what you chose to make it out of. Is the outside different with the deoxyribose and phosphate, or is it the inside basis?
The inside.
That's what makes us all different. So with our book, our genome, you inherited a gene that might say, it's like a recipe. And my recipe might make my eyes round. It might make somebody else's eyes really almond. It might make my eyes blue. It might make someone else's eyes brown, light brown, not even dark brown. And then there's a dark brown
Can I
gene.
ask you a question?
Yeah.
Why do people have different vision? I don't know how to say it.
That also is
Word. Because I'm tired of being blind.
nearsighted, farsighted is also
That's
a
why
gene.
I'm gonna beat up my dad or my mom for giving me that. 'Cause I can't see anything now. I can't see it. I can't see word or that word.
Yeah. Over there. Or you can't see them.
No, I can't see from right here. I know I can't see nothing. It's blurry.
Can you read up close?
Yes, doctor.
I know who's blind.
Daddy is blind.
Natalie is blind.
No. Wait, so what, how does you know how they laser your eyes, make your vision better? How's that work, Miss Thornhill?
Well, right now it doesn't have anything to do with DNA replication, so I can't answer it.
I wanted to ask a question that was important to my life.
I didn't break.
Elijah. Can you throw my marker? Is there a marker?
Excuse me.
Okay, so at this point, what we normally do is we draw this whole thing again. We're not doing that, but I want you to, let me make sure everybody understands my drawing otherwise. So see how this one has an old and a new? And this one's a new and an old. Something barred in blue, is that what you're saying? Each strand is identical. This one would be that one. This one would be this one. Mismatches or mistakes in the code. And this is what Wesley said earlier. So mistakes happen. Some mistakes are fixed, other ones are not fixed. I mean, I'm glad that not all of them are fixed. Otherwise, we'd still all look exactly the same. And it's not just the coolness of it. The other part is survival value. If a species is identical in genetic makeup, one virus could wipe them all out. Having differences within your species is a good thing. It actually adds your survival advantage. Okay, so survival advantage, differences, great thing. If you're not different within your species, not a good thing in terms of survival.
What time is
Okay.
it? It's 11:03 now.
Wow.
11:40.
All right, so what we're going to do before we get to, there is a set, you need one color in just a minute, but we're going to present our models first, okay? Or listen for mistakes. We hope you're going to present anyway, and they'll have to present on their own. You're going to present what you have, okay? All you're going to do is you're going to stand up, show us your model. Oops. That was decoration, I think.
She's broken.
Little decoration sitting on top.
Oh my god.
It'll be okay.
So you're going to hold your model. You're going to go through your key. Make sure that you and your partner both speak. Okay.
Can we go first?
And that's it. Okay.
Or are we starting now?
So what y'all need to do is you can put your markers or color pencils in your pouch next to you for now. Go grab this. Go grab your models. Uh-huh.
What do you call that part again?
Can I see if you're doing this?